Welcome back, everyone, to a brand new explainer. Today, we're focusing our lenses on the incredible microscopic dance of life. We are going to unpack the exact mechanisms of how biological structures actually grow, heal, and reproduce. If you've ever wanted a super clear step-by-step understanding of the cell cycle and cell division, well, you're in exactly the right place. Let's navigate through the blueprint of life itself. Okay, let's dive into this. Here's our roadmap for the explainer today. First, we'll uncover the secret of growth. Then, we'll break down the overall cell cycle. After that, we'll look at the two distinct types of division: mitosis, that's our equational division, and meiosis, the reduction division. Finally, we'll tie the whole shebang together by looking at why this matters to us on a macro level. Part 1, the secret of growth. To truly appreciate this, we really have to grasp the sheer, unbelievable scale of cell multiplication. Our source text poses a fascinating question right out of the gate. How exactly does a single cell become a massive organism? I mean, think about it. Even the absolute largest living creatures on Earth, a blue whale, a giant redwood tree, they all start their existence as just one single cell. Through a highly coordinated cycle of growth and division, that one parent cell and its progeny multiply into millions, billions, and ultimately trillions of cells. It's literally the ultimate biological mystery, and today, we're going to solve it. Moving right along to part 2, the cell cycle explained. So, to understand how cells multiply, we first need to understand their concept of time. In humans, a typical cell grown in culture divides roughly every 24 hours. But get a load of this breakdown. The actual physical division, what we call the M phase, only takes about an hour. That is just 5% of the entire cycle. The vast majority of the time, more than 95% of it, the cell is hanging out in what's called interphase. It's preparing, it's growing, and it's getting everything absolutely perfectly ready for that short, dramatic burst of division. And during that long preparation phase, interphase progresses through 3 distinct acts. First up is the G1 phase, where the cell just grows continuously. Then comes the S phase, or synthesis phase. Now, this is absolutely vital. This is where the DNA replicates itself. The amount of DNA doubles, say going from 2C up to 4C, but importantly, the actual number of chromosomes stays the exact same. Finally, we hit the G2 phase, where proteins are synthesized and the cell makes its absolute final preparations. Only after all of that prep work does it finally enter that tiny sliver of the M phase. Now, before we zoom in on that M phase, let's quickly demystify some of the essential scientific jargon so we have all our terms straight. The M phase actually happens in 2 distinct steps. First is karyokinesis, which is the physical separation of the daughter chromosomes, basically the splitting of the nucleus itself. Then comes cytokinesis, which is the actual division of the cytoplasm, cleanly splitting the body of the cell into 2 totally separate, independent daughter cells. Part 3, mitosis. Equational division. Mitosis is basically the biological solution to how an organism grows and repairs its body tissues, all while maintaining the exact same number of chromosomes as the parent cell. It's a flawless copying mechanism. And to achieve this, karyokinesis progresses through 4 highly choreographed stages: prophase, metaphase, anaphase, and telophase. While it's actually one continuous, fluid process, hitting these milestones helps us map out exactly what's happening to the DNA at any given moment. Watch how this choreography unfolds. In prophase, that tangled, messy web of replicated DNA condenses down into clear, compact chromosomes. Next, in metaphase, the spindle fibers attach and physically push and pull those chromosomes until they align perfectly right along the equator of the cell, what we call the metaphase plate. Then, anaphase kicks in. The centromeres split simultaneously, and the sister chromatids are literally pulled violently apart toward opposite poles. Finally, in telophase, those chromosomes arrive at the poles, they decondense, and a brand new nuclear envelope builds around each set. The nucleus has successfully cloned itself. Which brings us to part 4, meiosis. Reduction division. Now, if organisms are going to reproduce sexually by combining genetic material, they obviously can't just use standard mitosis. The chromosome count would just keep doubling every single generation. There's no way that works. Meiosis solves this by reducing the chromosome number by exactly half, and it does this in 2 separate cycles. The very first step, prophase 1, is incredibly complex and takes way longer than what we just saw in mitosis. It's so dense that it's broken down into 5 subphases based on chromosomal behavior: leptotene, zygotene, pachytene, diplotin, and diakinesis. Now, let's not get completely bogged down on the heavy terminology here. We really want to focus on the sequence of events. The chromosomes condense, they pair up with their homologous counterparts in a process called synapsis, and then something completely magical happens during that pachytene stage. So the absolutely crucial point is this definition right here, crossing over. While those paired homologous chromosomes are snuggled up really close, they literally exchange segments of genetic material between non-sister chromatids. They do this at intersection points called chiasmata. This enzyme-mediated swap is the absolute secret to genetic recombination. It's literally the reason you don't look exactly identical to your siblings. Right after that crossing over happens, the rest of meiosis I plays out. During metaphase I, those bivalent paired chromosomes align right at the equatorial plate. But notice the big difference here when we hit anaphase I. The sister chromatids don't split. Instead, the entire homologous chromosomes separate and move toward opposite poles. We've just effectively cut the chromosome number in half, creating a dyad of cells by the end of telophase I. Immediately after that, without any further DNA replication, the cells dive right into meiosis II. Now, this process looks remarkably similar to standard mitosis. The chromosomes line up at the equator again in metaphase II, but this time when anaphase II hits, the centromeres actually do split. The sister chromatids are finally pulled to opposite poles. Telophase II wraps the whole thing up, and we are left with a tetrad, 4 completely genetically distinct haploid daughter cells. Finally, part 5. Why cell division matters. Let's zoom out to the macro level for a second. How does all this invisible stuff dictate our everyday existence? When we compare these 2 processes head-to-head, the division of labor is actually pretty beautiful. Think of mitosis as the builder and the mechanic. It takes a diploid cell and makes an identical diploid cell. It's the reason a plant continues to grow from its meristematic tissues its entire life, and it's the reason the cells of your skin and your gut are constantly being repaired and replaced. Meiosis, on the other hand, is the legacy creator. It takes a diploid cell and reduces it down to haploid gametes, ensuring that when sperm and egg finally fuse, the specific chromosome number for that species is perfectly conserved across the generations. But hey, it is so much more than just conservation. As our source text states so powerfully, meiosis, quote, also increases the genetic variability in the population of organisms from one generation to the next. Variations are very important for the process of evolution. Because of that wild crossing over event in prophase I, every single haploid cell produced is totally unique. This microscopic mechanism is the direct driver of genetic diversity, adaptation, and pretty much the entire evolutionary history of life on Earth. Before we wrap up today's explainer, I want to leave you with a bit of a brain teaser straight from the chapter's exercises. Consider the highly coordinated nature of everything we've just learned. Can there be DNA replication without cell division? And conversely, can there be cell division without replication? Think about the exceptions to these rules and what that would actually mean for a living organism. It's a really fascinating puzzle that highlights just how incredibly precise the cell cycle has to be. I hope this explainer has illuminated the dance of life for you. Until next time, stay curious and keep exploring.